you for being here. Today, as a speaker, we have Marc Benavides from the Mediterranean Institute of Oceanography in France. Her expertise is in the study of the interactions between marine diazotrophs and their physical, chemical, and biological environment. And for that, she uses several combinations coming from nanotechnology, molecular biology, biogeochemistry, and modeling. Mar did the PhD in 2013 in Las Canarias, in Las Palmas de Gran Canarias, and since the PhD, she has made postdoc travels from many different places, including first in Germany, in fact. Then she moved to New Caledonia. She has been in Denmark, and since 2018, she's a permanent researcher in Marseille, in this institute. So she has come here also through one of these Severo Ochoa helps for visiting external researchers, and she's the first case of a scientist coming from this area. So we are very glad to have her here during this week, and she will present her work. Thank you.
Hello. Thank you all for coming, and for those connected online. And yeah, thanks to the Severo Ochoa Visiting Scholars Program for having me here this week. So I will, let me check. Yes, okay. So I will talk to you about nitrogen fixation and what we thought we knew and how things have been changing in the past years, and what is our current understanding of nitrogen fixation in the ocean. But first, what is nitrogen fixation and what's its role in biogeochemistry? Nitrogen fixation is the reduction of atmospheric nitrogen, which is usually inert to any other microorganism, to ammonium done by prokaryotes called diazotrophs. So these diazotrophs, they introduce reactive nitrogen into the system, which then can fuel primary production and also the export of organic matter, so it has an important role in climate regulation by the ocean. But where is nitrogen fixed? Well, nitrogen fixation itself, it's a very highly energetically costly reaction for the cell. So for each molecule of nitrogen, you have to spend about 16 ATP. And this is why we used to think that whenever you have nitrogen available in other forms, such as nitrate or ammonium, the cell will dare to fix nitrogen because it's much more expensive. So this is why traditionally nitrogen fixation research has been focused in the subtropical gyres where nitrate is usually below detection. However, when you put nitrogen fixation into the budget of the global nitrogen budget, and if you compare the sources of the inputs of nitrogen into the ocean to the losses, the balance between the two comes always negative. And this could either be because either the losses are being overestimated or maybe the inputs have been underestimated. And I'm going to try to convince you of the latter. This is the distribution of nitrogen fixation measurements in the global ocean until 2019. And you will see first that there is like a very a strong geographical bias. So most of our data comes from the North Atlantic. But also there's a regional bias because most of our data have been taken between 30 degrees north and 30 degrees south, so focusing in the tropics and the subtropics. So there's still lots of mysteries. We don't know what is going on in the Indian Ocean, we don't know what is going on in the Southern Ocean, not in the Arctic, and many places of the Pacific neither. So recently, studies have started to measure nitrogen fixation in unexpected places, such as the Arctic or in the Antarctic as well, and many coastal regions. This is just an example from the Eastern American, North American coast. And if you put all these numbers together, how much they think they contribute to the global budget, the global input could increase up to 20%. This number is of course highly uncertain because these measurements are very sparse in time and they are not systematically done. So we don't know to which extent we can really rely on this number. But more and more research groups are starting to be interested in measuring nitrogen fixation in unexpected places. However, measuring this nitrogen fixation in unexpected places also came with a surprise of unexpected diazotrophs or unexpected microorganisms. Traditionally, we thought that cyanobacteria were the main diazotrophs, and this had the advantage that more cyanobacteria are large enough and they are also pigmented, so you can easily study them just with traditional microscopy methods. However, with the advent of molecular methods, we have come to see that there is a really high diversity that is overwhelming in global databases of what we collectively call non-cyanobacterial diazotrophs, or NCDs, which are other types of bacteria and archaea that are not necessarily pigmented or photosynthetic. More evidence of this. This is a pulling of the data of the Tara Oceans expedition, and again, if you do metagenomes, you find as well data. Here they call it heterotrophic bacterial diazotrophs, but it's the same concept. They are overwhelmingly present in different size fractions of these samples collected across the ocean. Our challenge today is that actually these NCDs are not available in culture. There are very few representatives. This alpha protobacterium that was collected in the eastern tropical South Pacific, and there are some Pseudomonas strains collected from the Baltic Sea. But there is very little, few available examples in the lab that you can use to understand more their physiology and their metabolism. And if you put together all the sequences available today for NCDs, there are two main clusters that stand out, the gamma protobacteria and the delta protobacteria. And they also seem to be, have different distributions in different parts of the ocean. This is a recent example from my student Supadeep in the southern Indian Ocean. This is the tip of Africa, and this is Madagascar. And we could really see all the ASVs here in yellow. They are mostly protobacteria, and they are most abundant below 20 degrees of temperature, which corresponds to the southern part of the transect. Whereas if you go to the northern part of the transect, where the water is warmer, you again find cyanobacterial diazotrophs such as Trichodesma. If you go to more coastal places like estuarine waters, this is the Narragansett Bay in the eastern coast of the USA. We performed this monitoring along 5 months in different stations in this bay, and we found an overwhelming presence of delta protobacteria in this case. And we think that is because of this nitrogen balance in these regions, because most of these delta protobacteria can also dinitrify, so they can fix nitrogen but also dinitrify, so they are very versatile in these systems that have very high ranges of nitrate concentrations. And again, so also studying these non-cyanobacterial diazotrophs comes on top with the difficulty of facing a potentially different metabolism. So cyanobacteria, as I said before, they are pigmented, they do photosynthesis, so this potentially restrains them to the sunlit layer of the ocean, which is this very thin part at the most 200 meters deep. Whereas the others, they can potentially live anywhere. They cannot photosynthesize, so we think that they must depend on external sources of organic matter to obtain energy and carbon. And if they don't need light, maybe we can find them anywhere. So I became interested in studying nitrogen fixation in the deep ocean, and this is just an example from a cruise in the South Pacific. This is the Solomon and Bismarck Sea surrounding Papua New Guinea. And we studied, we measured nitrogen fixation rates between 200 meters and 1,000 meters. And together with the composition of organic matter to try to see if there was any link between the composition of organic matter and these NCDs. And what you see here is the nitrogen fixation rates in the bubble, so according to this reference size, and the oxygen in color, and you can clearly see that there was more activity in less oxygenated waters, which in principle made sense because nitrogen fixation is also inhibited by oxygen, because oxygen can deactivate the nitrogenase enzyme that these organisms use to fix nitrogen. So next we wanted to look at the organic matter composition to see if there was a relationship between this nitrogen fixation activity, these NCDs, and dissolved organic matter. So we use this method called FTI-CRMS, which can give you thousands of molecules for a given organic molecules for a given seawater sample. And this produces a wealth of data that is sometimes quite hard to summarize and to make sense of. But one way of doing this is to map your molecules in these bank prevalent diagrams where you confront the ratio, the molar ratio between the hydrogen to carbon molar ratio to the oxygen to carbon molar ratio, and this will give you regions of types of molecules such as lipids, proteins, amino sugars, and so forth. So we were very surprised when we did this with our samples that actually the organic matter that we found in the Bismarck Sea was actually totally different from the one in the Solomon Sea. And this was statistically correlated to the nitrogen fixation rates and also to the composition of the diazotrophic community. This is the composition, so this is a NIFH sequencing, and we found a high diversity, again, these gamma protobacteria, but also delta and others. But there was a very overwhelming abundance of these cluster 3 that are thought to be anaerobes. So we took just one example, like one of the most present sequences in our data set to design qPCR assays that would allow us to quantify these cells. And by doing so, we came up with this very strange result. So there's no particular ingredient either in depth or in distance. There is no particular relationship to the nitrogen fixation rates. So we wonder to which extent these communities can actually explain the rates that we are measuring in the dark ocean, or who else is fixing nitrogen there. So this takes me back to this plot because, again, we thought that cyanobacteria were mostly restricted to the upper sunlit layer of the ocean. But by the time we started to study aphotic nitrogen fixation, some evidence was coming out of cyanobacteria being present in the aphotic ocean. For instance, this is from the Malaspina expedition. There was just presence of Trichodesmium auricella, which are typical cyanobacteria diazotrophs, in many places around the transect. Also, evidence of sediment traps down to 1,000 meters and even more of depth, full with Trichodesmium. And also more recently, evidence from Tara, where you find other active diazotrophs such as using A down to 800 meters in transcriptomes. So we wanted to test this hypothesis, to which extent can sinking cyanobacteria from the surface ocean fix nitrogen at depth. And to do so, we did some trap experiments in a recent cruise in the South Pacific. So we had 2 stations with traps at these 3 different depths. And we used 2 tubes of these traps. One was filled with 15N isotope, so this allows us to measure nitrogen fixation rates. And the other one was filled with arenalator to preserve the nucleic acids. So we took the material from these sediment traps after 5 days of deployment, and we did nanosims analysis. So this is single cell mass spectrometry. And we found that they fix somewhere between 26 and 200 femtomoles of nitrogen per cell and day, which is actually very much in the range of what their activity they have at the surface. So it means that they are happily fixing nitrogen down deep. And actually, they represented some up to 70% of the bulk nitrogen fixation measured in the traps. We also sequenced NIFH genes from RNA in the traps, and again, we found that Trichodesmium, which is the green here, was the majority sequence found in the traps. Together with other groups such as Chlorobium, which is the purple here, or Jangia, the light green here, which are known to be typical epibionts of Trichodesmium, so they suggest that they sunk together. Okay, but now I'm going to go back to the NCDs because even if cyanobacteria can be present in the dark ocean, we still don't know what these NCDs are doing and if they fix nitrogen and how much or where. So we have now quite much evidence that these NCDs can be attached to particles. This is the abundance of diazotrophs across the North Atlantic, and the black dot represents some gamma A protobacteria. And you can clearly see that in smaller size fractions, so like a 0.2, they are not present at all, and whether you start raising up the size fractions to go to 3 microns or 10 microns, they do appear. So this suggested that they have a particle attached lifestyle. This was also shown by Fran recently, where both these gamma A's were found both in genomes and transcriptomes across the Terra samples in higher size fractions. But why will these particles be interesting for NCDs? Well, particles can be rich in carbon, rich in inorganic matter, but they also have they can have oxygen-free cores, so this would provide a niche for NCDs to fix nitrogen. This has been proposed recently in a modeling study where they consider the evolution of a particle with time and the evolution of oxygen within the particle. And the scenario would be that you will have a labeled particle in the beginning that is colonized by bacteria, which start respiring and then reducing this oxygen, which eventually would allow a little window in time where NCDs will be able to fix nitrogen. However, this has only been shown in models, and we don't know how this is really happening in true particles in the ocean. So we have started to play with this around in the lab. So this is my master student, Arthur. He has produced particles in the lab using phytoplankton cultures of diatoms and Synechococcus, and incubated them with or without glucose, with or without ammonium, to try to restrict nitrogen fixation. And indeed what he finds is that nitrogen fixation is higher when you have particles present, and also by labeling these NCDs, by labeling their nitrogenase enzyme, we can clearly see that, I don't know if the picture you see it very well, but they do show up only when particles, when particles are present, and not when particles are not there. And so something we want to do now is to try to understand more how this relationship between the particles and the NCDs happen. So we have started a collaboration with theoretical physicists to make videos of the cell trajectories to how they reach the particle, and to try to obtain models. And in parallel, because the particles can be large sometimes, we have started working on embedding them in resin so that they can be cut in different layers, so that you can do nanosims or mass spectrometry in individual layers to eventually produce 3D models of how this colonization happens. I'm sorry, but I don't have data yet, but very soon. So this takes me to my take-home message. Basically, I want to stand out that nitrogen fixation occurs beyond the tropics. It also occurs beyond the sunlit layer in different parts of the ocean, but we still don't know to which extent this is contributing to the global budget. I think that we have a huge need to develop high-resolution methods, because if nitrogen fixation in particles occurs just in a very small window in time, if you use traditional methods, very potentially you're going to miss the signal. And finally, that the full diazotroph diversity needs to be implemented in models, because global biogeochemical models today only consider cyanobacteria and just a couple of them. So thank you all. I would like to thank the program for having me here, Pep and Marta also for inviting me, and of course my group and all the funding that has made this possible. Thank you.
Thanks, Mara, for this presentation. We have plenty of time for questions.
Thank you, Mar. Very, very interesting. This is really nice to see all the, background and the new research you're doing. Very interesting. I'm curious about because we reconstruct some maps from the very deep ocean, from the bathypelagic, and I'm really dying to know a little more about what these microorganisms are doing in the body. So one of these potential diazotrophs that we found, it has the complete operon from the edge, and also have the rubisco. And they have two different copies, and they have also the pathways for the SOx oxidation, so to really capable in principle to capture energy with oxidation and sulfur. So and it was really more abundant, it was enriched in the particular types. And to me, this is the only one evidence so far that it was a chemiolytic autotroph of diazotrophs, at least on the genome level.
Yeah.
So this is do you know
that
any
would be
other?
so cool.
Yeah.
That would be so cool. Yeah, that's why I don't like when other groups call them HBDs, like heterotrophic bacterial diazotrophs, because maybe they are not heterotrophs actually. So the non-cyanobacterial goes well because that accomplishes. So all possible metabolisms. But yeah, that would be really cool. So I had something that I also had like this rather a hypothesis that it was an indirect pathway, so that maybe in their role in the dark ocean will be to provide chemiotrophs with ammonium to keep on going when nitrate has been already respired in the particles and there's no other nitrogen around. But yeah, maybe they do the whole thing just by themselves without.
Yeah, because I was wondering if we, because you have the possibility to label, no, the nitrogens, to try to capture this micro and see this.
Yeah, well, it's this antibody labeling that is tricky to make it work, but it's quite, yeah, it's quite easy to do. This one goes through to the traditional nitrogenase, the molybdenum, but I guess it can be also done for vanadium or iron. Nitrogenases as well as needed. We're also trying to develop alternative approaches of labeling, like using nanobodies, which are smaller, and that will allow like a faster traversing the membrane of the cell and avoiding complicated treatments and impermeabilization and so forth.
Okay. And the second question, now that I have the power, is because on Untitled, one of the results that was really also surprising to me was they found the diazotrophs in the fraction below 0.22 microns.
Okay.
So it was interesting because this is hypothetical, they can be, small, ultra small bacteria passing there, but it's that's they mapping the nitrogens and the nitrogen, and they found it high abundance in the viruses fraction, but because some bacteria can actually pass the filters. So but interesting is they found mostly in the Arctic.
Okay.
In the Arctic, in some mostly on the samples in the Arctic. So this is also, I think, another niche that they had to be more and more explored.
Yeah. Now the challenge is to measure the rates in these small guys.
Yeah.
Yeah. Very cool. Yeah, because in the Arctic it seems very like at the beginning they found UCNA, but I heard recently from other people that have been sequencing lately in the Arctic, and then it was full of deltas, no UCNA at all. So yeah, I guess it's variable, of course, depending on where you go, and also it's highly variable seasonally. So not to. Thank you.
I have a question. You started with the paradox to find nitrogen fixation in places where inorganic nitrogen is quite abundant in the surface of the ocean with the cyanobacteria, but then there is a second paradox, which is in the deep ocean where nutrients are even more abundant and there is a lot of nitrogen fixation. And I think I understood that this is linked to the particles. This is the total story. I mean, why nitrogen fixation should happen in the deep ocean where there is a lot of nutrients? So this is a paradox, but you didn't really
It is a paradox, but I would say because whenever you go, we will have rates from the Mediterranean, from the Atlantic, from the South Pacific. So but it doesn't seem to have a particular structure with depth and the relationships also with the composition of the organic matter, dissolved organic matter at least. It varies quite much. So it seems to be very heterogeneous. So this would be an explanation of why is it more related to particles, because particles can have a very heterogeneous distribution, but also because you find these NCVs in larger size fractions usually, and ecologically will make sense to them. But also, of course, why do they do it if there's plenty of nitrate around? But this is what I was just saying, that maybe at some point, if the bacteria and the particles have already respired the nitrate that is in the particle, at some point there's no more nitrogen around, so this will give some extra help to continue. That will be easier than respiring sulfate, maybe. But it seems that it's like a very particular window in time, or this is what models say, so now we have to prove the models.
And another question is, how do you sample the particles in the deep ocean? Because I have the feeling that the particles are very soft in general, and very unstable, no?
Well, it depends. There are many types, like fecal pellets are very solid, but sometimes it's very fluffy material. So either with marine snow catchers or sediment traps or in situ traps, or different approaches. But yeah, indeed, manipulating them now that we are trying to embed them in resin to do the microtom cuts, and yeah, most of the times you destroy them when you try to.
So, and my expertise is that when you analyze bacteria from the deep ocean, is that all of them look very well dispersed, very nicely dispersed. But then the signal that we get from the molecules, it seems that particulation is very important. But what we see in the microscope is that everything is, we don't see particles in fact, in the deep ocean. Looking at the Malaspina samples, I didn't see particles. Basically, very well, nicely dispersed, the bacteria.
Hi, congratulations for your work, Mark. I like a lot. I would like to know, how did you measure or estimate the DOC liability? Because you said that you related with the diazotroph, so how did you do that?
In this study, only up to 1,000, and then in the Mediterranean, I think we went down to 2,000 or something like that. But yeah, you know that this FTICRMS kind of misses the most labeled part, so this is not the...
No, but you relate the DOC liability with the diazotroph, no?
Mhm.
So how did you know the concentration of DOC which was labile? Did you do incubations, or how did you?
No, not really. Like here it was quite indirect, so we were just looking at the composition of the different, like if we had more amino acid like molecules or more protein like molecules and so forth. But we have also done experiments in parallel where you add sugars or you add amino acids or you add phosphonates, and yeah, they always respond to something, and so they like that you give them organic matter, but I'm not sure still to which extent it's because of the energy or because of the carbon.
Okay.
And it's very variable, depending on in different places, in different studies. We have a preferential response to sugars, and then sometimes a preferential response to amino acids, and sometimes to phosphoesters. So it's super variable. So yeah, I don't have a super conclusion yet. I also think that when you do this kind of experiments, of course you give them something very like Coca-Cola, no? So you give them like this particular compound that you have bought from Sigma, and it's. So we are trying now to produce organic matter in the lab, labeled with 13C, so that we can trace it into their biomass using mass spectrometry methods. But it will be like a more natural matrix composed of something more realistic of what they will find in the ocean, because of course glucose, it's a very small molecule, they take it up and it disappears, but it's not realistic how much glucose is there really in the ocean that you can, like as a stock, it's just cycled super quickly.
Because couldn't you do like some incubation with so in situ, so when you go to take your samples, then you can get that water also incubated to see the DOC, which is labile, and then correlated with. I thought that you did that, but
no. This is what we want to. We're going on a cruise recently, and this is what we want to do. So we're going to add DOM labeled with 13C, and follow it over time, how the DOM changes and how the community responds.
Okay,
so
nice.
I'll let you know.
Looking forward for the results.
Thank you.
Hello, thanks for the nice talk. I was wondering, I'm not super familiar with D-nitrogen, so I was wondering what are the sources of D-nitrogen in the ocean, and what is the pattern worldwide, what makes it change the concentration?
Mm-hmm. Well, it's very stable actually, because so this makes like some 70% of the atmosphere, and it's just diluted dissolved into water as any other gas. So it's always in excess. N2 is never limiting actually. Well, unless you go to OMCs and other more complicated systems, but in general in open ocean waters it's in excess. But diazotrophs are the only ones able to use it.
Hi, thank you. A great talk. I had a question that goes in the line of what Ramon was saying. It's food for thought. So It appears to me then that this nitrogen fixation is not so much related to a bulk nitrogen limitation in the ocean at all, but to the micro environment around particles, right? And this would give the paradox that you would have more nitrogen fixation in areas that are already rich, like upwellings, coastal shelves and so forth, where you have lots of particles, lots of micro anaerobiosis around particles and so on. So it has nothing to do then with a bulk nitrogen limitation of the ocean, which is something really interesting, I think.
Yeah, exactly. The moment you have more particles or more organic matter, you're gonna have more nitrogen fixation. This is what we see also in estuarine places that are really rich and full of nitrate. But even when in the summer, when you have a lot of nitrogen limitation, is when these NCDs come up and when you see more nitrogen fixation. So there is like this balance. But yeah, food for thought, indeed.
Well, thank you. And I was wondering also where phosphorus limitation kicks in then.
Yeah, we know that they love phosphorus. The moment you add phosphorus to any of these incubations, they are totally happy. But it's always it's more that the ratio with nitrogen that really matters. Because even if you have a lot of nitrogen, as long as the ratio with phosphorus remains low, then they are happy. But the moment you have the opposite situation, then it doesn't work as well.
Thank you, Mark, for your wonderful talk. I'm very intrigued about the relationship between oxygen and nitrogen fixation. And I'm wondering whether in your colonization experiment, if you plan to track the oxygen evolution during the experiment, if there is a way to measure that, or?
Yeah, we have started doing so but just like the surrounding oxygen in the free living fraction, so to say, so just in the incubation water, and just track it. Mostly worried because we are accumulating lots of particles in a small volume, so you're inducing a lot of respiration because of course these cultures are not axenic. But it would be cool to go with a microprobe way and really see what's inside the particle and how that changes indeed. But what I'm thinking is how to do it visually, something you can see in the microscope.
Yeah, because you were mentioned also that you're planning to embed the particles in this material, so wouldn't it be easier to put the probe there, or?
I don't know. What I know from people that do this kind of stuff, like Morten Iversen and so forth. So they keep the particles in suspension by applying a flow the other way around. So they keep the particle in suspension all the time, and then they insert these really tiny probes into the particle to get like gradients of the concentration of oxygen from the exterior towards the inner core. But yeah, maybe stabilizing them in these gels. Well, it depends. Like the gels we use to make the cutting. So this is epoxy, so it's really hard. But before that, you do a pre-embedding in agarose, which is still soft. So yeah, maybe why not? I guess it depends on how stable the probe is, because apparently it's super easy to break them.
Okay, thank you.
In the data that you show, you study nitrogen fixation at different depths of the water column, and I had the impression that the amount is the same or similar in the very deep ocean than at the surface. And that's very strange because the biomass of microbes is so much different between the surface and the activity level, no?
Yeah.
So do you have a clue for that?
Well, I will say it's a bit like what happens with the biological carbon demand. So that they have like more per cell at the end, I would say, because when you this is the tricky part as well, when you calculate nitrogen fixation rates using mass spectrometry methods, the equation, it's normalized by biomass. So you're multiplying by biomass. So the moment you have more biomass, necessarily you're going to have a higher rate. But only if you take the relative flow rate, just like the difference between the isotopes, you have this higher difference sometimes in the deep ocean, but it's so sparse, like there's no particular pattern, and it's hard as well because because there's little biomass, you have a lot of limitation with mass spectrometry because you need like a minimum to get into the detection limit of the machine. So we're talking about incubations of 20 liters per replicate to do this. So sometimes it's really limiting. And of course you have the problem of the light, the problem of the temperature, and maybe the pressure as well. So it's tricky to set up these incubations.
Yeah, I just forgot one more question. You mentioned that you found nitrogen fixation in the sediment traps in the mesopelagic, right? And there was a trichodesmium, you mentioned, but for how long? So I mean, could be just very recent, particles sinking from the bulk. But maybe the tricky question here, for how long can be these guys still fixing nitrogen?
This is what we don't know, because of course we fill the traps with 15N labels, so the moment one trichodesmium or anybody gets inside and it starts fixing, but what you collect is at the end of your trap deployment, which can be 4, 5, 6 days, 1 week, or whatever you want to do. Of course the experiment was not planned for this, I just took advantage of the deployment to do something on the site. But yeah, we don't know exactly when. You only collect at the end, and of course you divide, when you calculate your rates, you normalize for 1 day, and you deploy for 5, but maybe they were just fixing for 1 day, and actually the rate is much higher. So it's very uncertain as well.
But I think that there might be experiments done with trichodesmium because it's in culture in dark.
Yeah, we have also done. We have in the lab these chambers where you can simulate the pressure. So these are like titanium bottles where you increase the pressure and decrease the temperature at the same time, and we have done so with cultures of trichodesmium, and it also works. And then to try to explain why, we have been trying to work on a metabolic model to try to explain how much carbon they have when they begin to sink, and how long can they survive only with this source. With this source of carbon, because there's no light anymore. So.
Just related to Sylvia's question, what do you think that would happen with trichodesmium, for example, in the dark ocean, in terms of the diel cycle for nitrogen fixation? Trichodesmium fixes during the day, and so do you think that could keep fixing nitrogen the whole time, or will they keep the diel in the dark ocean, or?
I guess so, to some extent, because cyanobacteria are very stubborn. I don't think it has. I don't know to which extent it has been shown with trichodesmium, but at least with I remember some old papers from cyanotiki, and even you put them in complete darkness for like a week, and they keep on trying to photosynthesis and nitrogen fixation opposely during the day. So I guess that just as unless they don't have any more energy, and then it's done. If they cannot synthesize their enzymes anymore, then it's over. But how long that lasts?
But you think it will keep the cycle?
Yeah.
Okay.
I would say so, because they are really stubborn, old.
So another take-home message is that cyanobacteria are very stubborn. Any other question? So we don't have many questions. Thank you again for your
Thank you.
cooperation.